The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone. Welcome to today's webinar that will focus on Flex-FPM for single cell manipulation. My name is Edward Nelson. I'm a technical sales specialist at Nanosurf based out of Boston, Massachusetts. Online also is Patrick Fredericks. He's in Liestal, Switzerland. Will be taking your questions. If you have any questions to ask him, you can use the chat button or the question button. And we'll collect all those questions from you, and at the end of the presentation we will we'll be able to answer them. Patrick, are you there?
Yes, I'm here.
Okay, good. So today's webinar will be on Flex-FPM. Flex-FPM stands for FluidFM Probe Microscopy. So it features FluidFM is a multifunctional nanomanipulation tool. It features hollow AFM cantilevers. These are connected to a pressure controller. The cantilevers come pre-assembled in what is called a cytoclip, and here's a sort of a diagram of that. That connects the cantilever itself to this pressure controller in a really easy to use system. These cytoclips are mounted to the Flex AFM scan head, and this confers all the positional accuracy and force sensitivity of a modern research-grade AFM to the system. So the entire Flex-FPM system includes the hardware platform, which I described, the Flex AFM, which is up here on an inverted microscope, the microscope itself, pressure controller, this is the AFM controller, and a vibration isolation table. We also have a software that integrates all the hardware together, and a touchscreen with an intuitive user interface. Finally, the last thing I also mentioned was these FluidFM probes, which are on this cytoclip. The FluidFM probes come in a variety of different styles. There are tipless cantilevers. These have 2 to 8 micron openings. These are used for eukaryotic cell adhesion or cell sorting experiments. We also have apex opening cantilevers. These have 50 to 600 nanometer openings. These would be good for something like lithography or for bacterial adhesion applications. Finally, we also have these syringe tips, which have openings on the side. These can be used for cellular injection or extraction experiments. All the tips are manufactured using standard microfabrication techniques. An SEM image, like the ones you see here, are provided with each of the tips, and customized options are also available through FIB. So with the cytoclip, it's very, very simple to mount the cantilever. If any of you have any AFM experience, you know how tedious or how much practice it takes to load a cantilever using tweezers. Here, a media, it's probably blue food coloring, but nonetheless, media is put into the back end of the cytoclip in the reservoir. These cytoclips come in these sterile packaging. Notice we don't have to use any tweezers at all. We can just lift them out with a gloved hand. They clip directly onto the cantilever holder. There's no tweezers necessary. On the left here, or on the right here, excuse me, is the AFM scan head. Now we just take the cantilever holder, and what we'll do is attach now the tubing to the back end, and it attaches nicely with a lower lock. Then there's 3 magnets that snap the AFM or cantilever holder right into place. That's it. As I mentioned, the Flex-FPM is an AFM, so it has all the functionality of a research-grade AFM. An AFM measures force using a cantilevered probe. Probe deflections are detected using a laser beam, and beam deflections are proportional to the force. In imaging mode, deflection information is a reconstruction of the sample topography. This is a near-field technique, so you get information from your sample that is beyond the optical resolution limit of your microscope. The FluidFM cantilevers or probes are microfluidic control system. The other part of it is a high performance pressure controller. It's computer controlled and uses a closed loop controller to maintain constant pressure on the microfluidics. You can apply pressures from negative 800 millibar up to 1000 millibar gauge pressure on the cantilever. There's a switching time of 10 milliseconds. That allows precise deposition of material at the tip. This video on the left demonstrates how one can deposit material with the hollow cantilever and pressure controller. This cantilever is filled with a fluorescent molecule which lights up as it exits the tip. Another component of the system is the inverted microscope. This gives you a bottom-up, unobstructed view of the sample. It also preserves the epifluorescent path, allowing correlated fluorescent and forced microscopy. So on the right you can see an example where you have GFP expressing yeast cells and unexpressed cells on the same image. And we can also measure the force there. The last aspect is the software in the system. So it integrates all the hardware components within a single software platform. It uses an innovative user interface with a touch screen and pre-designed workflows. For example, if you wanted to image, you can do things like change the scan area or move it around to anywhere on the screen using your finger. You can move the sample by just dragging your finger across the screen. When you start actually collecting an image, you notice that we're building up an actual topographical map on top of the bright field image, and that makes it easy to correlate the two images together. So Flex-FPM truly is a versatile tool. In this talk, I'm going to just concentrate on these four applications: single cell adhesion, single bacterial adhesion, colloidal spectroscopy. So this is spectroscopy using a bead like colloid, and finally single cell isolation. There's also, I mentioned with the syringe tip, you can do injection or extraction of a cell. You can also spot or do nanolithography even if the surface is under another liquid. Because these are totally enclosed cantilevers, you can spot under another liquid that way. There are many, many more applications, and I encourage you to visit our website on the applications section, and there we have some papers and some application notes on some of the other methods that I'm not going to cover today. So the first part of this webinar, we'll talk about single cell adhesion. So for example, in a workflow, here we have a HeLa cell on a fibronectin surface. In the workflow, we select the cell using the crosshairs. The cantilever goes to the cell, and now the sample is brought up into contact. Under pressure is applied, and as we withdraw the sample now from the surface, we are measuring the force on the cantilever, and we get a force distance curve as we see here developing. Now notice the sample is out of focus. When we remove the cantilever, we see that the cell has been detached from the surface. So we know that it's now stuck to the cantilever. So that's basically what a single cell force or adhesion measurement would look like. Here are two other representative force curves. On the left is a force curve for Candida albicans. It's on DDP, or a hydrophobic dodecyl phosphate. This is a dimorphic yeast cell. It's well-studied in human pathogen, or a well-studied human pathogen that is a threat to immunocompromised patients. I'll describe a little bit experiments with this cell in a later slide. On the right is the HeLa cells that we saw in the previous slide, or previous video. It's on fibronectin. So what we want to take, the kind of data we want to take from this is the maximum adhesion force, shown here, the distance from the surface in which detachment occurs, and if you take the integral here of this, or the area under this curve, you can get the work of adhesion. And all those three bits of information can inform you about the binding affinity between the cell and the surface. So onto a specific example. So C. albicans exists as two phenotypes. So in other words, it's dimorphic. When these yeast cells transition from the oval to the hyphal form, this is a sign of an infection. This is sort of what a hyphal form looks like instead of this nice round ovals. The transition can occur after adhesion to a solid surface at sort of the conditions of the body, so at 37C and sort of in the right media. It's been found that the protein HGC1 has been identified as this essential protein for this hyphal morphogenesis. But the question is HGC1 involved in the actual adhesion mechanism? That's what we want to try to find out. So by using an inverted microscope, the authors in this paper could measure both cell types at the same time, or one after the other, on the same identical surface. So that way they know the surface is identical for both systems. The image here, as I showed it before, is the C. albicans. The wild type is actually expressed in GFP, so it's going to come up green. And the mutant strain is the unlabeled one here. This shows a modified HGC1 protein. What they found was that the wild type yeast exhibited strong adhesion at 37C. So as you let them incubate on the surface, after 350 minutes, the adhesion force steadily increased. The modified cells, though, on the other hand, always showed low adhesion for all times. This suggests that the defect in HGC1 not only keeps C. albicans from transitioning into hyphal form, but also modifies the adhesion properties of the cellular surface. And I just want to point out again that any questions that you have, please enter them into the chat box, and we will, at the end of the talk, we'll try to answer as many as possible. So moving on, I want to talk about now these endothelial cells. So in terms of human eukaryotic cells, biomechanical or biomedical engineers have a lot of interest in how cells stick or don't stick to engineered surfaces. In general, a medical device that is inserted into the body will cause inflammation. This is known. For example, vascular stent, here's an image of one. It's inserted into blood vessels, may lead to clotting. This is called thrombosis, or may lead to a blocking of the lumen, the lumen here. It's called stenosis. A stable cellular interface between the implant and the body regulates tissue homeostasis. Or body equilibrium. So in vivo, this function is performed by the endothelial cells. Now, the strong substrate topography strongly regulates the function of the endothelium. So, So what is the adhesion force between the endothelial cells and an engineered substrate? So we're trying to design a biomedical device, insert it into the body, and we want to understand how the endothelial cells bind to that engineered substrate. So endothelialization is initiated by integrin membrane receptors and ligands on the substrate. Attachment triggers recruitment of the cytoplasmic proteins, these focal adhesion kinase and paxillin. These phosphorylated kinase catalytically activate cytoplasmic packs and leads to a mature focal adhesion. So only mature focal adhesion can apply a force through actin filaments. The engineering strategy for medical devices then is to generate surface structure that affects the relay between the packs and the kinase and the paxillin. So in this case, they are going to look at structures with different groove depths from a flat surface, not shown here, 100 nanometers all the way up to 1 micron grooves. So experimentally, the question becomes, what is the adhesion force between endothelial cells and an engineered substrate? Now, what's something that they could do? So there are other commonly used adhesion assays. For example, a washing assay. So the cells are on the substrate. This is the red here is the substrate of interest. You just try to wash the cells off. You can use a spinning disc in which you just try to spin the cells off. Laminar flow, so in a microfluidic, apply a laminar flow across the cells, or centrifugation. You're trying to force these cells off the substrates. These all apply a force, true, but the force is a shear force mostly, or sometimes always. And these are all bulk techniques which are not quantitative and lack reproducibility. On the other hand, FluidFM is a single cell technique in which the adhesion force can be measured quantitatively. We can measure forces from the tens of piconewtons up to hundreds of nanonewtons. We have good control over the contact position, such as the applied force and the time in contact. Because of the pressure controller, we have strong adhesion to the cantilever within seconds. And because the cells are not attached chemically to the cantilevers, we can detach them, so we can do serial experiments with a single cantilever. This is both time and cost effective, and it gives us more statistics to work with. So back to this experiment, what the researchers did is they used a Flex-FPM, and they used this FluidFM micropipette with an 8 micron aperture, a spring constant about 2 newtons per meter. On the right are some representative force distance curves. Here is a force curve for a flat surface, all the way up to a force distance curve for a the surface with a 1 micron or 1,000 nanometer grooves. And what they found was that for the shallower grooves, so 100 nanometer grooves, they found stronger adhesion force, and for deeper grooves they showed a weaker adhesion force. Now the flat surface had a similar adhesion to the deep grooves, and here this is a surface that was treated with blebistatin, which is a small molecule inhibitor. This is shows affinity for the myosin II skeletal muscle. Then they compared the results that they got from the single cell force spectroscopy with a different technique, laminar flow migration. So they watched how the cells moved in a laminar flow. They found that cell motility was lowest for the shallower grooves. Let's see if I can zoom in here. So they found that cell motility lowest for the shallower grooves, and so cell motility is really an indication of immature focal adhesion, and it shows a decrease in PAX and an increase in the focal adhesion kinase. For the shallower grooves, performance is lower. Okay, let me get back to this. All right, so combined, these results indicate that the communication between the kinase and the paxillin is affected by surface topography. Okay, they also found that the cell adhesion correlated with the cell contact area. So if you took an image of what it looked like with between the two substrates, the two different types of substrates, they found that for deep grooves, the cells can just bridge the groove gap there. Whereas for shallow grooves, the cells would grow into and out of the grooves. So what they found was that if a cell could grow into the grooves, so follow the topography of the surface, they had a higher adhesion force. Whereas if it bridged the grooves, it had a much lower adhesion force. So that in itself was pretty interesting. Okay, so I want to now switch up to talk about single bacterial adhesion. So bacterial adhesion is a process that affects cell physiology and survival. In other words, the survival of many forms of bacteria depends on efficient biofilm formation. So what is a biofilm? Biofilm is a 3D structure of cells that's embedded in an extracellular matrix. It starts with a single cell attachment and grows to a large mass that then disperses to form colonies elsewhere. So quantification of the detachment forces, these initial detachment forces, facilitates understanding of the process affecting cell physiology and survival. So, for example, in order to manage the Staphylococcus biofilms on medical catheters, understanding bacterial adhesion is the first step. So I've already mentioned some of the bulk methods to characterize cell adhesion, and they're not all very quantitative, right? So that you would use essentially the same sort of bulk techniques. The AFM offers a way to measure the forces directly. What happens though, you have a single cell and a single cantilever. So you can attach that cell to the cantilever through various methods, receptor ligand, electrostatic, glues even. But what happens is it's time consuming and it's costly. A better solution to this is, again, FluidFM. With a single cantilever, you can look at many, many different types of cells. The cell is immobilized to the cantilever with under pressure and released with overpressure. That means you can do serial measurements and have higher throughput and more statistics. But bacteria have to be handled differently than mammalian cells. The shape and size of the bacteria, and they also have smaller forces. So for this experiment, they used a pyramidal probe with an apex opening, 600 nanometer apex opening aperture. They also used a cantilever that's a little bit softer, about 10 times softer than the one we previously showed. The image sequence shows a typical measurement. First, the cell is identified. The cantilever is moved into position and brought into contact with the cell, while an 800 millibar under pressure is applied to the cell. Then as we lift the cantilever, we see that the cell detaches from the surface, and notice the neighbor is still there. And applying an overpressure, the cell can be removed and another bacterium selected for measurement. So these are, I guess, the results. Single-cell force spectroscopy of E. coli shows, unsurprisingly, I think, that the adhesion force depends on the contact time. So if you come into contact with the cell and press it into the surface for 0 seconds here versus 60 seconds, you'll get a much different adhesion force. The same can be said as the amount of force that you push the cell into the surface. So in the first case, you push down with 1 nanonewton of force. If you increase that to 20 nanonewtons of force, you can see that there now is other effects from the membrane or from flagellum that are changing completely what the contact or the adhesion looks like. Notice that this is out into microns from the surface, whereas when it doesn't have time or enough force to bind to the surface, it's only in 150 nanometers from the detachment occurs. Overall, the higher or the bigger, the longer the set point, this generates a tighter cell substrate contact up to a point. And no matter how much you wait, you won't get any more adhesion force after that. So these look like they're reaching a plateau. So the advantages of Flex-FPM for SCFS versus conventional AFM. So you have longer cell substrate interaction times. The cells can be left on the substrate to incubate until you're ready to start the experiment. Higher cell substrate adhesion forces. So the force on a cell is a function of really the aperture size. So with a 8 micron aperture, you can apply forces up to almost 3 micronewtons. That means larger adhesion forces between the cell and the substrate are measurable. With suction, you can also attach. With the suction, cells can be attached to the cantilever in much less time than conventional AFM, in seconds versus minutes. That means you can do more adhesion measurements in a shorter amount of time. So, for example, the time it takes just to calibrate or to mount a cantilever and then calibrate, because you can pick up and release a cell, you can do a lot more measurements. Whereas in a traditional method, you would need to mount a cantilever, wait for it to bind to the cantilever, and then do the measurement. Then you have to either clean the cantilever or get a new one and start again. So in the course of 30 minutes, you can do 6 measurements, whereas it would take almost 90 minutes to do 2 measurements with conventional AFM. So for this next part, I want to talk now about the colloidal spectroscopy. And so in conventional AFM colloidal spectroscopy, putting on a bead on an AFM cantilever is a multi-step process. First, the bead and the cantilever need to be coated with an antibody, or first the cantilever is applied, or glue is applied to the cantilever. You stick a bead to the cantilever, and then you need to apply a antibody or chemical to the bead, which is also a multi-step process there. After that, you conduct your experiment, but if you ever pick up any dirt or debris from the cell body, or if the chemicals on the bead do not become ineffective, you have to throw away the entire cantilever. With the FluidFM system on the Flex-FPM, with a tipless cantilever, you can pick up a bead and then do your experiment. And as soon as you pick up any sort of debris, you just apply a positive pressure and remove that bead and then grab a new one. These beads can be pre-coated. You can use smaller beads. Remember, we have a 2-micron aperture tip, so you can get much smaller beads possibly, and it's a very simple exchange. This means more data can be acquired with a single cantilever, saving both time and money. So what are some applications of colloidal spectroscopy? Well, one is elasticity screening. So if you pick up a bead, so for example a 4.5 micron bead, you can measure the contact mechanics between the cell and the bead. From that you can measure, or you can determine the elasticity of a cell, or you can measure other nanomechanical properties. You can also coat the beads with antibodies or other chemicals, as I mentioned. Then you can measure recognition sites on cell surfaces. But you should realize, though, that the interaction decreases with subsequent measurements. So one study found that after 10 measurements, the adhesion force decreased to 60% of its initial value. So the ability to exchange beads quickly and efficiently is critical for consistent results. Alternatively, though, instead of using beads, you can use cells instead of the colloid to measure the stiffness of the cell directly. So in a recent study published just this year, a group measured the elasticity of these oocytes with the Flex-FPM. So these cryptosporidium oocytes are waterborne protozoa that have been implicated in water plant contamination incidences. For example, in 1993 in Milwaukee, there was an outbreak that was responsible for 104 deaths. The Flex-FPM can be used to measure the stiffness of single cells and thereby identifying pathogenic versus non-pathogenic cells. The way it works is, again, you have a field of view where you can see the actual oocysts. The cantilever selects one, or you select a cell, you pick it up with the cantilever, and then you measure on a hard surface the elasticity, or the spring constant of the cell. So far from the surface, there's no contact. We make initial contact with the cell, and then we have hard contact with the cell. And from there, you can get the stiffness of these oocysts. So in the work that was just published, what they did is they measured the height and stiffness of the two cell lines. So the pathogenic form of the Cryptosporidium parvum in gray has a different, though overlapping height, that's down here, the height, versus the sort of benign C. murus. But if you look at the stiffness is also overlapping, but they are separated there between the two. So they found a separation in height and a stiffness between the two cell types, and this could be used to identify the pathogen from a mixed sample. Now, because of this reversible binding, they were able to measure 30 cells per hour with 8 measurements per cell. This gave them the ability to get a lot more data from in a short amount of time. They also looked at treated versus untreated C. parvum. If you were just looking at the size of the cell, you notice that just the height, that you can't distinguish between untreated cells and those that have been heat treated or freeze-thawed treated. But if you look at the stiffness, there is a clear difference between the untreated cells and the treated cells. The difference is even more clear if you look at just the histograms here. For the untreated, it's shifted to the right, and for the treated cells, you see that there's about almost a 1 Newton per meter softer there. At 30 cells per hour, one could conceivably use this technique to identify pathogenic oocysts in environmental samples. In fact, that's part of the problem of traditional methods of detection, such as filtration, purification by immunomagnetic separation, immunofluorescence assays. All these cannot discriminate between infectious and non-infectious oocysts. So this may be a viable technique to get around that. The last thing I want to talk about in this webinar is single-cell isolation and cell sorting. I showed you before how you can actually pick up individual bacteria. So with a pyramidal cantilever, you can select and remove individual bacteria. So this might be interesting if you were trying to do genetic cloning and you want to select a single bacteria to do the cloning on that single one. A more interesting is manipulation and sorting of mammalian cells. These are cells that even on that are tightly bound to a surface, that have been cultured on a surface. So what you can do is fill the FluidFM probe with something like trypsin and then locally deposit that trypsin right over the cell. So for example, here's a Hiehest cell. It's a fluorescing red red fluorescent protein. We bring the cantilever just above it, and then we release trypsin, which is also marked with this cascade blue marker. After a certain amount of time, the cell detaches from the surface, and notice that none of the other cells are affected. We can then target that cell, pick it up, and move it somewhere else. If we now do more analysis, if we were just trying to sort those cells. The benefit here is that we don't have to apply trypsin to the entire culture, and that reduces the stress on all the cells. In the fluorescent microscope, we now can sort them by fluorescence or by any characteristic that you want. So in conclusion, the Flex-FPM is a technique that combines the positional accuracy and force sensitivity of an AFM with the picoliter position of the FluidFM system. The instrument can be used for quantitative and serial measurements of adhesion forces between cells and surfaces, and to directly manipulate cells to measure either the stiffness of the cell or to isolate and remove cells from a confluent layer for further analysis. I hope you enjoyed the webinar and learned something new about FluidFM for single cell manipulation. Before we get to the questions, I also want to extend an invitation for all of you to attend our Flex-FPM and FluidFM user meeting in Würzburg, Germany on July 4th. The meeting will feature talks and discussions by current FluidFM users and will include topics on single cell manipulation, spotting and nanolithography, and protocol discussions on development. It is for current users and future users alike. If you would like to attend, just send an email to info@nanosurf.com and we will provide more details. Thank you. Now, I think Patrick is still online, and we'll try to answer some of your questions.
Yes, I'm still online. We got quite a lot of questions, so I don't know whether we will be able to address all of them. I mean, one of the first questions that came in was on instrumentation. We showed an active isolation table, but how is it with acoustic isolation? Can you comment on that?
So in my experience, if you want really fine force steps, if you're really looking at small forces, you definitely need the vibration isolation. Let me go back to... So if you notice in this slide here, there are very small force jumps here in less than 50 piconewtons. You definitely need, if you're looking for information such as this, you definitely need the vibration isolation. In my experience, I haven't needed the acoustic isolation, but it can't hurt. And Patrick, if you want to comment on that too?
Yeah, it strongly depends on the laboratory environment. If a laboratory is very quiet, you might do without, but then also one should stay quiet during experiments. Otherwise, an acoustic isolation definitely helps suppressing the environmental noise.
Yeah, actually, a lot of one of the benefits of acoustic isolation also is blocking airflow, which sometimes is actually more critical to your measurement. You'll get drift or things like that. So that's an added benefit of acoustic enclosure too.
Okay, another hardware-related question: the accuracy of the pressure controller.
So it has 1 millibar accuracy.
Okay.
I believe, yeah, so you can set between negative 800 millibars to 1,000 millibars with 1 millibar, plus minus 1 millibar accuracy.
Okay, then more, yeah, consumable side, the spring constants. What is the spring constant range, and what are there are a few questions also on the force resolutions you can get with the hollow cantilevers.
So right now the two types of cantilevers have a nominal spring constant of 2 newtons per meter and 0.2 newtons per meter. So for bacterial adhesion experiments, I would recommend the softer cantilevers, and with those you can get these small steps that you see here.
Yeah, so actually for the micropipettes, that's the tipless cantilevers with the large openings, they also have a 4 Newton per meter cantilever, so a bit stiffer, so that you can even get higher forces, adhesion forces measured. Mm-hmm. Related to that, what are the limitations for cell sizes to be measured with single cell adhesion experiments?
That's depends, I guess, on how big a cell. I mean, the cantilevers are around 40 microns across. The largest aperture size is 8 microns. The limit, I suppose, is really what the cell attachment to the surface is. With the larger apertures, you can apply a 2.4 micronewton theoretical, I guess, force to the cell. If it's such a large surface area, I mean, if you're talking about a 50 or 60 micron cell on the surface, then you might have trouble applying enough force to pull it off the surface. Yeah, if Patrick, if you want to add anything to that?
Yeah, so I guess on the small side, bacteria with sub-micrometer diameters have been measured. Getting much lower, you might run into problems that the opening is getting so small that you cannot apply sufficient force. On the large size, at the moment we are limited also by the Z range. We have an external Z actuator on the sample side which has 100 micrometer piezo range. That means if you need more than, say, 80 micrometers to pull the cell from the surface, you also run into problems, and that puts an upper limit to the cell size you can measure as well. Yeah, it's also related to this. What is the piezo range?
So yeah, so the Flex has a 10 micron piezo range, and as Patrick just mentioned, you can also get the system with a 100 micron Z range, and that's what we saw in the video of this Hiehest cell. So the let go. So the notice that when you bring the when you start the force curve, the cell goes out of focus. So the cantilever remains in the optical path or the focus of the microscope, and then we move the sample away from the cantilever. So that allows us to measure the adhesion of larger cells or very sticky cells too.
Okay. Let me see what other questions were there. How can you see whether detect whether the beads are fouling if you do colloidal spectroscopy?
That's
a tricky one.
Yeah. If you're looking at it sort of an adhesion force, you'll start to notice that you're not getting any adhesion at all. It's really something that you can't use them forever. So it's the same with imaging, I suppose, that at a certain point your data doesn't sort of match what you expect, or wasn't the same in the end as it was when you started. And so at that point you have to consider either cleaning or recoating the cantilevers, or with our system just getting a new bead. And because it's so easy to exchange beads, you can do 2 or 3 measurements, grab a new one, and do it again, and without any loss of productivity.
Yeah, I guess for elasticity mapping, where you would like to suppress any adhesion, you can just measure until you see that adhesion is getting significant, and then you know it's time to get a bead, a new fresh bead with a fresh anti-adhesion coating on it.
Mm-hmm.
Let me see, go a bit through the other question, I guess. That how can you get a tight seal between an object and a hollow cantilever?
Well, if you're, for example, the E. coli, which are barrel-shaped, in that case, it's probably a good idea to actually use an FIB to mill out the shape of or what you expect the shape of the E. coli to the cantilever. And this is actually what they did in this paper. That way you have the area around the cantilever or around the aperture fits with their surface, and you form a much better seal.
Mm-hmm. Okay, let's go for a final question. We cannot treat all questions, but we can come back to you after the webinar to answer the remaining questions. One question that always comes up, not only now but also before, is cell viability. How does the under pressure affect the cell membrane, and also have we checked cell viability with FluidFM experiments?
I can answer the first one. So suction will affect the cell, but so will a chemical binding to a cantilever. So what's nice about it, that within 5 seconds you attach the cell to a cantilever and you're ready to measure. Where for chemical binding methods, it takes upwards of 20 minutes for any adhesion to take place, and that can affect signaling, cascade signaling in the cell can affect a lot of things in the cell. So we're applying it's a physical force, I suppose, to the cell versus a chemical force, and we do it in a much shorter amount of time. And so we believe that the effect on the cell is minimal. But maybe Patrick, if there's been a study done about that?
I know that on the oocyst paper that was mentioned, they looked a little bit at the suction force, how it affects the measurements. In general, yeah, I would say yes, the under pressure does affect the experiment, as does any other method, and one would have to do comparing studies to see which effect is most problematic for the cells. I think for some experiments the under pressure is not an issue. There will certainly also be cells where under pressure might cause a problem. As for cell viability after experiments, I think if you're talking about the bacteria, I don't expect any influence there. Yeast cells, I know they have been picked up and put them in other position, and then you could follow this the yeast budding afterwards, so they easily survived. If you pull off a mammalian cell with micronewton forces from a surface, you will certainly damage a lot in the cell. And I don't think there have been structural studies on the cell survival after such experiments. Okay.
So if that's questions, I think we can answer the rest of them offline. We'll contact you directly, I suppose. And I thank you for your attention. And please, if you have any questions, you can refer to the email or the info@nanosurf.com if you have other questions beyond what you've seen here. And look out for new webinars in the future where we'll try to look at other applications possible. Patrick, any last words?
Yeah, by saying anything I get last words, but no, I don't have anything to add.
Okay, so thank you for your attention, and we'll see you next time. Bye-bye.
Bye-bye.